welcome to our on-ground NEURON 2022. We're so happy to see you. Last year's virtual program was successful, but it's really nice to see everyone in person for today. So my name is Dr. Adrienne Betts, and I'm going to do the introduction. I'm going to let you know a little bit about who helps organize this conference before I introduce our keynote speaker. So this conference has really grown over the years and is quite successful. I thought I heard that feedback here. And I just want to take a moment to tell you all about how much sort of effort goes into this wonderful program every year. So I do need to start by thanking our new dean of the College of Arts and Sciences at Quinnipiac. He generously donated some funds so we could continue this conference this year. Again, my name is Dr. Adrienne Betts. I'm at Quinnipiac University, and I direct the Behavioral Neuroscience program here. Dr. Haight is upstairs running down, or he was here a moment ago. He's helping. We're simulcasting upstairs also, in case people need to move around. Dr. Ahern, he does a lot of the program and sort of the background behind NEURON, so he's not a front face, but he is so critical to this mission. And then UConn, we allowed UConn or asked UConn to help sponsor us, and so I'd like to highlight a couple individuals, including Renee Rotolo, who has since moved on to, she's academic to industry at this point. Alev was our poster judge 2 years ago and is now a really wonderful coordinator. We have Dr. Tanner, who's here in the front. He's been involved in NEURON for a couple of years, and Elizabeth Klein is a new organizer as well. And then there's some other individuals from UConn. So I'd like to just thank everyone for this wonderful effort. Okay, and so a couple years ago, we migrated to the Eventbrite platform. It's been very useful for us. We thank you for registering this way. This is a moment to say if you have not checked in, we do really need you to check in, because this is a way we keep track of who attended and from what university. And speaking of that, I need to give a little shout out to 3 universities who have brought so many students here: Mount Holyoke, Holy Cross, and Skidmore. So I think I might, at the end of the day, go run a report and see who gets an award, because that's really amazing. And those are some distances. So thank you so much for coming every year and supporting NEURON. And so as we've grown in the last, say, 12 years, our geographical, we've broadened, so it's really impressive how far you come and the commitment to sort of giving neuroscience away, and thanks again for coming from wherever you came from today. So a couple reminders. So this program is the hard copy. It printed midweek, so there's some changes. So if you want the most up-to-date version, you can use this QR code from a phone, a tablet, a computer, and there'll be the updates there. Please don't download this interactive program. We should have QR codes all around campus today if you need more information, but this is not as up-to-date as the interactive guide. I am not going to go through this entire schedule of events. I'll do that after, Renee Rotolo will do that after the keynote, but I just wanted to let you know this is where we are at this moment. We're in the keynote with a welcome in the keynote, and then we have some really wonderful programming for the rest of the day. This year's keynote is being recorded, as always, and we will post it on our Quinnipiac YouTube channel. You have to go into YouTube and then search Quinnipiac and get to our Quinnipiac YouTube channel, and then you can see all the historical NEURON keynotes dating back from 2011. So now I'm going to take a moment to introduce our keynote speaker, Dr. Sanacora. So Dr. Sanacora is an MD PhD. He received his PhD in physiology and biophysics from State University at Stony Brook, and his MD in 1994. And then he moved to Yale, and he completed his internship at the Yale New Haven Hospital. His current role there is he's professor and director of the Yale Depression Research Program. His research largely focuses on the pathophysiological mechanisms of depression, highlighting amino acid neurotransmitter systems in particular. So his interest in the neurobiology of mood and depression and antidepressant action will be of really interest to us today. He uses he's a translational researcher, so he does basic science and rodent models. He looks at chronic stress, inflammation states, novel treatments for major depressive disorder. He has a clinical laboratory as well where he does fMRI and spectroscopy. Excuse me, I'm being recorded, so I can't say the word that I normally say all the time. And so today's talk, the aim is to show the progression and the evolution of the research that has led to S-ketamine gaining FDA approval as an antidepressant agent. So this is very timely. We're really excited to have him talk with us today. There'll be some time for questions and answers that will be led by Dr. Tanner after, so be sure to get some questions ready. A dialogue is what we're looking for here. And please welcome Dr. Sanacora.
Thank you all. Thank you, Adrian. It's a pleasure to get a chance to talk to you all. The presentation I'm going to give may be a little bit different than what you're typically used to hearing. I'm really going to try to take you through a story, a timeline of how ideas really from the earliest stages come through to the point where it actually reaches FDA approval, and the final step is actual clinical implementation to being used clinically, which is, I think, what we all hope for as translational neuroscience professionals. So as I said, I'm hoping that we can cover these three things, but really the main message I want you all to get is kind of the story. Don't worry so much about the details, but the big picture of what it's like to take something from the basic science lab and to bring it to actual clinical utility. I consult to many different pharmaceutical companies, and this is my disclosures here. But to start with, this is what many of us all hope to do, is to really bridge that translational gap. The idea of taking something from basic science, and for most of you, from basic neuroscience principles and bring it to clinical practice. And this paper is now about 10 years old, but I think it still holds very true. You know, they call it the Valley of Death because it's really hard. It's hard to get across from these basic science ideas to actually making it to clinical practice. And there's been a lot of resources, a tremendous amount of resources that's been put into this. And you can actually see the rate of increase of funding, R&D expenditure, but the amount of money going to PhD training and PhD work compared to MD or MD PhD. But if you look over here, the rate of actually new molecular output is not that great. So, you know, it really makes us question, you know, how we're putting our money and how we're doing this research. And one other thing I really say, I'm going to start out with almost a negative thing, but hopefully we'll spin this into a real positive. And something that I think should really be almost mandatory reading for anybody going into the field is this book by Richard Harris. He's actually one of the science writers for NPR, or was one of the science writers for NPR. And it really highlights the real pitfalls, the dangers of trying to do biomedical research and where things can go wrong and how much money can be spent without a real productive outcome from it. So I really recommend everybody at least read through this at one point. It's a quick read. Not to, and very clearly he states the purpose of this isn't to discourage people. It's actually to highlight where the pitfalls are and to explain how, and to try to have people think about how to do this better. So this is kind of this big challenge. How do you go from this academic, preclinical neuroscience model, for us neuroscience, but this is for all of science, to actually bringing treatments, which is usually done by pharmaceutical industry. And here they list a bunch of steps where things can go wrong. And you can read these. These are all kind of the classic ones. I add one, just having poor foundational hypotheses to begin with, which I think is really critical. You have to, you don't want to build a big temple on a house of cards, on a foundation of cards. You want to make sure that you have good foundational hypotheses that have been well tested before you start to move on. But you can see there's many spots along the way, and one including having actual clinical relevance. When you're starting to do work, if your goal is to have a clinical treatment at the end, you want to make sure that it's going to be relevant before you start. So hopefully with this talk, we'll go through what are some of the lessons that we learned directly from the development of ketamine and S-ketamine, and highlighting some of these specific areas here where I feel like we've learned quite a bit. So if we start with the idea, what is the clinical relevance? So I'm not sure for all of you if you realize, but major depressive disorder, which is what ketamine had been developed for, is actually determined by the World Health Organization to either be the #1 or #2 leading cause of disability throughout the world. That's kind of shocking to many people. They're not really aware of that. In terms of death, suicide is actually in the US the 10th leading cause of death for all ages, but below the age of 42, it's usually about the second leading cause of death. So we're talking about a serious illness that has tremendous morbidity and mortality associated with it. And the scary thing and the humbling thing is the rates are actually increasing, especially in the US. So there's clearly a need for new treatments when you're talking about clinical relevance. And this was a study done called the STAR-D study that was finished about 15 years ago, looking at the existing medications at that time, including cognitive behavioral therapy, and trying to look at it through a sequence of treatments. And we do pretty good at treating people. We can get the large majority of people into remission. The problem is if you didn't respond to one of the first 2 lines of treatment, the chance of responding to the 3rd or 4th line dropped dramatically. And this is largely because we had one type of medicine. It was the monoaminergic drugs. So the TCAs, MAOIs, SSRIs, SNRIs, they all targeted very similar mechanisms within the brain. So it wouldn't be surprising if you didn't respond to the first or the second that you're not going to respond to the 3rd or the 4th. So there was really a need to develop new treatments for depression. And the last thing was, it was just accepted at the time that treatments for depression took weeks to months. If you were suffering from depression, you started on a medicine that may or may not make you actually feel a little bit worse when you first start taking it, but you tried to convince somebody who had extremely low motivation to begin with because of their illness that you had to take this for weeks or months on end before you're going to get a benefit. And it was just accepted. This is just 20 years ago, standard literature. This is it. Nothing can work faster than that. So we really, there was a need to develop better treatments that work by a different mechanism, and potentially treatments that could work quicker. So what was the rationale for using ketamine? I don't know how many of you heard of ketamine, and if you have heard of ketamine, at least 10 years ago, 20 years ago when I started doing this, people would typically, if they heard of ketamine, unless they were an anesthesiologist or working with ketamine in rodents, they thought it was a horse tranquilizer or a party drug. But now I think people have heard a lot more about it. But what's the basic rationale for trying ketamine? And this goes back, and this is a cardiology slide, this goes back to the idea that if you look at the pointer here, I think I can actually just do it with this. No, I can't. If you look at the rate, this was the age-adjusted death rate due to myocardial infarction, due to heart attacks. And if you look at it from the 1950s going up through the mid-1960s, early 1970s, the rate of age-adjusted death due to heart disease was actually increasing over time. And that had gone back for years. But if you look right at this point, so I'm going to try to do this, hopefully, there you go. Right around there, you see this major inflection point, where all of a sudden we made a huge difference in the late 1960s, early 1970s, compared to if the rate stayed steady or if it continued to increase at the rate it was increasing. And you start to ask yourself, well, what happened? What happened in that period in the late 1960s that changed things? And most people would argue we really started to understand pathophysiology and the risk factors associated with heart disease. So we started to understand how important it was to keep your blood pressure in control. So better antihypertensive medications came out, better antihypercholesteremic medications started to develop, antiplatelet coagulating drugs started to come out. And then also people really started to realize, well, the major risk factor was smoking. If you could just get people to stop smoking, you'd make a huge impact. And you can see that dramatic benefit that came about. With depression, we really didn't have that. We really didn't have a great way of saying, well, what is the primary pathophysiology behind depression? What is the primary risk factor? Over years, and this is a very recent paper we just published in Nature Reviews, But it was known, and now there's more and more evidence supporting the fact that stress we can have a 3-hour lecture just trying to decide what the definition of stress is. But if we think of stress in the general term as something that's really perturbing you from a homeostatic set point, especially cognitively and emotionally for this purpose, can have deleterious effects. And we learned a lot more about that. This, a little bit of stress is actually a good thing for most people. If you look at most measures of brain function, it usually improves somewhat with stress to a certain point. And that could either be longer duration of stress or too much intensity of stress, or the other factor is loss of perceived control. But when you start to combine all these, especially at critical points during development, you start to have more neurotoxic-like effects of stress on the brain. And that seems to be one of the major risk factors, and probably if we understood the pathophysiology associated with that, we could start to develop new treatments that could actually either act prophylactically to prevent the onset of depression and other neuropsychiatric illnesses, or actually treat them. So there were some seminal studies that came out in the late 1990s. And some of these were work by Wayne Drevitz and others who actually started to show, really for the first time, that there were true metabolic differences in specific brain regions of patients suffering from depression. The anterior cingulate cortex became a primary spot where we could show there was real abnormalities in metabolism, but even going one step further, looking at the actual architecture and looking at the brain regions and the gray matter within that brain region, you can show that there were differences in people that were suffering from depression and healthy controls. And then you're able to show other brain regions like the hippocampus, very similar findings. Taking it to the next level is actually able to show some cytoarchitectural changes. And again, I'm sorry, this pointer has stopped working, but you can see over on the right is some evidence that glial cells, surprisingly, the neurons fairly normal. There's really not great evidence of neuronal damage in depression coming out, but there was increasing evidence, at least in the time, this is the late 1990s, suggesting that there were glial cell abnormalities occurring in these brains. And the interesting thing was, we could show in rodent models that we can produce very, very similar effects by stressing animals, like specifically chronic unpredictable stress type models, or even intrusive stress type models. You can get very similar effects in the brain. And also at the time, started to really understand how stress could be a key factor in the neurobiology of generating some of this neurotoxicity. Work by Bob Sapolsky, and many of you may have known Bruce McEwen, who was at Rockefeller, really started to do a lot of work to show specifically how stress can have toxic effects within regions like the hippocampus and prefrontal cortex. And this idea that it may involve what's called excitotoxicity, where increased levels of glutamatergic drive, coupled with problems in energy metabolism, and higher levels of some of the stress hormones like glucocorticoids can come together to actually cause this toxic environment in the brain that can lead to maladaptive changes in brain function. Other work, actually from Beta Mogan at Yale at the time, could show that glutamate was large bolus release along with acute stress. So you see this large increase in glutamate, the major excitatory neurotransmitter. And then a lot of work by Dennis Choi and others showing how this actually could lead to toxic events with higher levels of glutamatergic stimulation. So this is all happening in the late 1990s, and this is paired with some of the work, Phil Skolnick, who was at the NIH at the time, who started to get this general idea that when you stress animals, there was good evidence of impaired LTP. And it was evidence that this is stress was impairing neuroplasticity in one sense that could be measured by LTP, and showing that if you started to target the NMDA receptor, the glutamatergic NMDA receptor with some drugs, you could have some effects that look like antidepressant actions in rodent models. So this is all occurring in the late 1990s when everybody, if you said depression, serotonin, or norepinephrine, those were just synonymous. Nobody was thinking outside of that box at the time. My own personal entry into the field, was doing some magnetic resonance spectroscopy work, really more interested in energy metabolism than anything else. But we kind of stumbled across this finding that patients suffering from severe depression had very low levels of cortical GABA and abnormal levels of glutamate, depending on where you looked and how you looked. So just some more evidence suggesting that there was abnormalities within the brain associated with these amino acid neurotransmitter systems. And interestingly, the amino acid neurotransmitters are very tightly coupled. In fact, the primary cell in mediating the amount of glutamate and GABA is the glial cells, especially the astroglia. So really a story started to come together for us is that, if there are some of these stress-related impairments in glial cell function, could this be related to some abnormalities that we're seeing in the depressed patients in terms of glutamatergic abnormalities, but also in terms of how this is affecting neuroplasticity. At the time, it was fortunate as people started to say, well, maybe these amino acid neurotransmitters are important, maybe glutamate's important, and specifically maybe the NMDA receptor of glutamate is important. Is there a way of testing that? And at the time, the drug ketamine was out. So ketamine, really interesting story, developed in the 60s, FDA approved in 1970 as an anesthetic. It really became very popular in use in field hospitals, especially during the Vietnam War. This is a very safe anesthetic. Unlike most other anesthetics that bottom your blood pressure and heart rate, decrease your respirations, ketamine doesn't do that. So it became a very safe anesthetic to use, and it's still used throughout the world, especially in third world countries that don't have the same OR setups that we have here. But Ed Domino, I put up his picture, he just died a few months ago, was really one of the first people that looked at ketamine and started to understand the mechanisms of how it was working, specifically its role in the NMDA, on the NMDA receptor. So we had this drug, ketamine, that could be used, and there was increasing thought that, well, if the glutamatergic system is involved, could we start to play with that? So my colleagues at Yale at the time, John Crystal, Dennis Charney, Rob Berman, and a few others, John Crystal had been doing work for years with ketamine, looking at it as a model for psychosis, looking at the role of glutamate in schizophrenia and other psychotic disorders. So he had a lot of experience with it. And then when this hypothesis started growing, the idea that the primary damage or the primary pathology within mood disorders and depression seemed to be in regions of the frontal cortex and hippocampus and not in the pons or in the raphe where really the monoaminergic systems are primarily present, the idea was that maybe we should start thinking of glutamate in some way. And this is actually a quote taken right from John at the time. If one viewed depression as a disorder of cortical limbic function, then glutamatergic and GABAergic signaling would be implicated. This perspective shift led to the test of the NMDA glutamate receptor antagonist as a probe for glutamate signaling. So it was really this sort of shift from thinking that we have this simple serotonergic, noradrenergic deficit hypothesis to thinking more of impairment in neuroplasticity and the role the glutamatergic system and GABAergic system may play that led to this idea. And it was a very simple study that was done with 8 people, really tiny study. And this was just giving ketamine at 0.5 milligrams per kilogram, which is a sub-anesthetic dose. There was no more thought given to it except for the fact that that was the dose that was used when they were studying its effects to mimic sort of psychotic thinking, where patients would have these changes in cognition and perception, but still stay awake so they can report it. Nobody was trying to find the right dose for anything. It wasn't that it's hypothesized that this was the dose. It was just arrived at that way. And the way they actually did the study was 1 week they came in. This was done at the West Haven VA, not too far from here. They were treated either with the 0.5 milligrams per kilogram IV over 40 minutes or given IV saline. And you can see this is the Hamilton Depression Rating Score on the left. This is a measure of depression severity. Going down means you're getting better. And remarkably, you can see within hours, people were getting better from their depression the day they got the ketamine. Getting remarkably, dramatically better compared to when they got saline, really no improvement at all. And this was really quite amazing to everybody. And again, a quote from Dr. Crystal here is really to the amazement of our patients and ourselves. we found that ketamine produced rapid, profound, and surprisingly durable antidepressant effects that were temporally dissociated from the brief acute behavioral effects of the drug. So ketamine, very short half-life. So within an hour or two, the acute effects of ketamine are gone, it's metabolized, and you pee it out, it's gone. So you don't really having much of an effect at all. But the antidepressant effect continues to go over time. So, now the drug is gone, but yet people are continuing to get better, lasting days at least after, which is really novel. It took 6 years for that to go to the next step, where at the NIMH, Carlos Zarate and Dennis Sharma were able to replicate. If I showed you this, the graph on the Hamilton Depression Score would be identical to what you see there for the study done at Yale. But this here, I showed the amount of people having a response. So you can see within 1 day, 71% of these people that had treatment-resistant depression that had not responded to other treatments, were responding to ketamine within 1 day. And you can see it lasted for a few days and went down. If you compare that to what would be 8 weeks of treatment with the standard oral antidepressants, like Wellbutrin or Effexor or the typical SSRIs, so within 1 day you're getting a response that would be equivalent to what we would typically see after 8 weeks of treatment with these other medicines. So a really pretty remarkable finding. That was 2006. That led to this very quick flurry, and it's all relative terms. Quick meaning, so now we're up to 2015. And you can see the studies that were done after this, giving a single dose of ketamine and looking at the odds ratio of response. And you can see about 10 to 1 odds ratio of having a response at 1 day to ketamine compared to placebo, which is really kind of unheard of or extremely rare for something like depression. Even with these small N studies, these are relatively small studies. And then even if you look for a week out, you still see over about a 6 to 7 odds ratio. So really pretty remarkable finding. But these studies were all done a single dose and in really small N's. I think the largest N here is less than 50 people in any of these studies. And now you're 15 years out. 15 years have gone by to this point. But with that, once those studies started to come out, it caught media attention. And I think this speaks in part to the desperation. People suffering from depression was really desperate for new treatments, and their family members were desperate for new treatments, and their clinicians, the psychiatrists, were really desperate to offer new things. So this idea that ketamine may have this rapid antidepressant effect really caught fire, and all the major media outlets picked up on it and started presenting this. And it actually got to a point where the clinical use got pretty far ahead of the actual data. And we actually did a survey back at the time just looking at the growth of how clinicians were starting to use ketamine. And you can see from 2006, when that first replication paper came out, to 2016, there was this huge increase of people offering ketamine to their patients. And the number, we estimated, was over 10,000 people easily treated with really minimal data. And most of this data was just a single dose, showing that it lasted for a few days, no evidence of prolonged effects and no evidence of safety. So this was an interesting time in science to see how things can the cart can get ahead of the horse a little bit if you're not careful. And that led to the American Psychiatric Association asking me and a few other colleagues to try to put our heads together and say, all right, well, what do we know about the safety of this drug and where are we? Because we're really balancing this great unmet clinical need with relatively limited data. So we were able to put together a consensus statement that we couldn't really give great advice because we didn't have great studies, but we could kind of draw boundaries about, all right, well, this is what we know, this is what we don't know, these are the questions that still have to be answered. And I think that was quite helpful in sort of steering the course and where to move next. And we highlighted several different things. So there were some immediate clinical relevance, like what's the optimal dose? Remember I said that first dose was chosen really because it was a dose that caused some cognitive and perceptual effects and people stayed awake. Really no more thought given to it than that. We didn't know much about the long-term effectiveness or safety at all, which is, when you're going to start treating a patient, something you really do want to know. And then we knew little about the moderators of the effect. So could you use it with other medicines? Is it dangerous to use it with another medicine? Are there certain people that are going to respond better or people that are going to respond worse? We knew very, very little about that at the time. And we knew very little about the long-term safety. We knew that people that abuse ketamine, because ketamine has been a drug of abuse, can actually have toxic effects on the brain, on the bladder, on different organs. So this isn't a completely safe drug you're giving. So then we went back and as groups started to go back and say, well, what is the optimal dose? And here's a little bit of back translation. So going back from the clinic back into the bench, there was, went back to the rodent lab, and this was Ron Duman's lab. Ron was at Yale and unfortunately passed away last year or 2 years ago now. But his lab was able to use the forced swim test. Very weak. So use the forced swim test, which I don't know how many of you know this, the forced swim test. It's a very high-tech model. If you put a rat in water, you see how long it swims for before it gives up. But it still is one of the best predictive models we have of antidepressant-like effects. And the interesting thing is, Ron was able to show that at these sub-anesthetic doses, like 10 mg per kg is sub-anesthetic in rodents, in rats specifically, you get this nice antidepressant-like effect, meaning the rat swims longer. It doesn't have the same level of despair, or at least it doesn't give up. But if you went up to 80 mg, which is like more of an anesthetic dose, we're looking 24 hours after the dose. So it's obviously you're not putting the anesthetized rat in. It's 24 hours after you get it. But they don't have that antidepressant-like response. So it's pretty amazing. And then now something a little bit more believable, to me at least, than the forced swim test is starting to look at some of the cellular mechanisms. So is it?
It's good, just to get a little more.
Okay,
Thanks,
some of the cellular mechanisms. So Ron was able to look at some of the enzymes, phosphorylation of some of the critical enzymes in the mTORC-C1 pathway, which is really involved in controlling local protein synthesis. So as this work stimulated a lot of basic science, we started to understand that it seemed like the ketamine was rapidly increasing plasticity and spine density changes, which critically involved the mTORC-C1 pathway. So interestingly, able to start to see the phosphorylation, you start to see activation of this pathway, moving up from 1 milligram to 5 milligrams to 10 milligrams. Even if you go to 30, you get even a bigger bump in that. It's not on here. But again, if you go to 80, if you give that anesthetic dose, you lose that effect, which is really interesting, suggesting that you have an inverted U-type curve, and that it's what's called the therapeutic window, that you have, it isn't going higher is better. It may be a therapeutic window where you can actually go too high to lose that effect. We were able to use some NMR spectroscopy work to show that the glutamate-glutamine cycling seemed to be critical. And it was the same type thing, that as you increase this dose going up, you get this increase in glutamate release. So it looked like what ketamine was doing, it blocks the NMDA receptor, but by blocking that NMDA receptor in some way, and we can talk about how, actually causes more glutamate to be released. So you start to see glutamate being released and turning the glutamate-glutamine cycle quicker as you go up until you get to 80. Where it actually stops that. And again, we were able to replicate that same thing with the forced swim test, that if you go too high to the same point where you stop this increase in glutamate release, you stop the increase in mTORC-C1 activation, you no longer are getting this antidepressant-like response in the rodents. And this matched actually older data using microdialysis. So I'm not sure how many of you are familiar with microdialysis. It's actually a way of putting some silastic tubing right in and measuring the efflux that's coming out. And you can see that these sub-anesthetic doses in the rat, 10, 20, 30 mg per kg, actually cause this increase in glutamate release that very much matches this idea that glutamate is being spilled out. But if you just double the dose, you get to 50 mg per kg, you shut off that glutamate release. So it seems like there's a critical component there. And it seems like for some reason that releasing glutamate was really critical to getting that antidepressant-like response in rodents. How does it actually work in the clinic? So this is just an idea of how you try to do this translational work. This was a large study sponsored by the NIH. This was 5 sites, a collaborative group, including us, MGH, Mount Sinai, I think. I'm trying to remember who else was part of this. But 5, I think UT Southwest and maybe University of Alabama, we all got together with this and tried to run this study where we tried different doses, 0.1 mg, 0.2 mg, 0.5 mg, or 1 milligram per kilogram of ketamine. And it was compared to midazolam, which is a rapid-acting benzodiazepine, which was used as the placebo, as the control for this study. And interestingly, you can see, and I really wish my pointer was working here, but it's not. Yeah, oh yeah, there we go. There we go. Yeah, the trackpad's a little wonky. So this was the 0.5 mg per kg. And this is 20 patients per group over these 5 groups. So the 0.5 was here, the 1 mg per kg was here, suggesting that by going up, you got no great added benefit to that. And then, interestingly, the 0.1 was here and the 0.2 was there. Oh, great, thank you. So, the bottom line was the 0.5 looked the best, but to be honest, this is one of the lessons to everybody. Even with 100 people in this study, we didn't have the power. We didn't have the statistical power really to say anything definitively. I think we could say definitively that ketamine did better than midazolam as a whole, but in terms of the individual dose, it was fairly limited. So the next step was even just figuring out how frequently to give it. So this is a study actually sponsored by Janssen Pharmaceuticals, part of a division of Johnson & Johnson, where just to show, well, what if you keep giving it? Will people stay better? Because up to that point, most of the studies have just been giving it once and people relapsed. There was one small study at Mount Sinai that gave it over 2 weeks and kept people better, but nobody knew what dose. Nobody knew how frequently to give it. So this is a study where people were randomized to either get treated twice a week or 3 times a week. And then within that, they were randomized to either get the saline placebo or to get the active ketamine, 0.5 mg per kg. And from this study, you could see, and you can look at the responders and remitters, that we could see that twice a week was just as good. Now, this study had, about 16 to about 15 to 16 people per group. These are still really pretty small studies, but there was enough from this to suggest that there was no benefit in treating more than twice a week. And that really served as the platform to move forward. So then the question is, what is the optimal route? So giving IV ketamine is great, but not many psychiatric practices are prepared to give IV. It's really, most don't have infusion pumps. Most aren't familiar with using IVs. Most don't have the infrastructure needed. So Janssen, the pharmaceutical company, tried to set out to find a way of delivering this intranasally. And they thought to do that, they would have to give a more potent version of ketamine because of the volume to be used. So what was determined was that S-ketamine, just the S enantiomer, has a much higher affinity than the R for the NMDA receptor. And based on that hypothesis was the idea that if you used S-ketamine alone, you could use smaller volumes because it has a higher potency. Does that make sense to everybody? Now, that being said, it also was a way of ensuring that you had some patent protection. You can look here and see that the S-ketamine has about 4 to 5-fold greater affinity for the NMDA receptor. And with that, the first studies were done saying, well, what if we use low-dose IV S-ketamine? And that was a study sponsored by Janssen. You can see it did really well. Going down to 0.2 mg per kg did really well with IV. Quick dose, people did very well. And they moved forward with that. And then they moved to the intranasal version. So now it no longer had to be given IV. Now you can give it through an intranasal spray. And they were able to do some dose finding. Now you can see sort of the sample size here, about 30 in each group there. And for the follow-up, only about 6. But they showed clear evidence that compared to placebo, this intranasal was working. So this is all great, but we're still looking at sample sizes of less than 100. And I know for some of you right now, you'll say, I wish I could have a sample size of 100. But I can tell you in the real world, if you go back to, the rigor mortis book, we have to be so careful interpreting data with small n. And in clinical trials, 100 is a small n. So what happens when you move forward? Now, this is where big industry comes in. So this is where Johnson & Johnson comes in. Now they set up this series of what's called phase 3 studies, where you can see there's 346 people in the one study, 223 in another, 138 of people just over the age of 65. And then there's one with 802 people that took the drug for up to a year to get the safety. So now these are real numbers that give you real idea of safety and efficacy. And just as just to give you some sense of what this costs. So to develop a drug, the average cost right now is somewhere expected between 1.4 to about $2.83 billion. That's a billion. That's how much money it takes to do this type of studies. To really get something to the point that you can get it to the FDA and say, this works, this is safe. Just to put it in perspective to total NIMH funding, they get about $2 billion a year to fund all their programs. About 1.5 in 2020 will go to research, extramural research grants. So it would be like taking all of your money that's funding 3,000 or more research projects, centers around the country, and just putting it to developing one drug. Just put it in perspective that way. It's a very expensive endeavor to get into this. Just very quickly, it's really unique study design, and I don't want to take too much time, but what the Janssen actually did was take it where people had to have proven not to respond to an antidepressant at first, and then start a new antidepressant, prove not to respond to that. And then uniquely, people were randomized to get treated either with a new standard oral antidepressant, either an SSRI or an SNRI, or the placebo. Everybody got the new standard oral antidepressant. And then on top of that, they either got S-ketamine or saline. So really unique study design. And this is the data. Now you start looking at large number samples. So this is the first, which is called the fixed dose. So they either got 56 or 84 milligrams. And you can see here that it's pretty reliable. I mean, you start to see what you're getting. This was the placebo, which the placebo was added onto a new antidepressant. So this is how people did with new antidepressants. This is, again, looking at the MADRS, which is another measure of severity. And moving ahead, now you look at that data, it looks great. Most people, I have so many things here. It looks great. That actually did not hit statistical significance according to the a priori statistical data and analytic plan, the way that it was set up. You can look at the response and remission rates, pretty impressive, but it missed just slightly. The second study, which was the flexible dose, meaning they can get dosed with either 56 or 84 milligrams, looks very identical. And again, so the nice thing about studying hundreds of people is it's replicable. You can do it again and again and get very similar findings. This was statistically significant because they didn't have 3 groups, they just had 2 groups. Very clear. And then if you look at the response or remission, which clinically is actually the most important thing, we learned 2 things. One, they're getting 60, 70% of people having a response and getting over 50% of people having a remission. These are people that had not responded to any of the standard oral antidepressants in a very rigorous way. But you did look at the ones that were treated in this protocol with a standard antidepressant. You're still getting remission rates of about 30%, which is twice what would have been expected if they were just being treated like in the STAR-D study clinically. So we learned 2 things. One is that there's a big benefit of participating in clinical trials, and 2 is it actually does look like the drug works. So I just want to move ahead, and this is the study of 800 people treating people up to a year. And you can look at how people got better and stayed better. And another lesson of large numbers, you can see how flat that looks. Those are error bars there. Very little error. I can tell you not everybody did well and stayed well. People are bouncing around here, but you add 800 people together and you get pretty small error bars. And I'm going to skip through this. And then is it safe? So we looked at all of our data for Yale, studying people for years, and we had 14 people that were treated for a long term with it, and it was pretty safe. I can tell you from safety data, 14 people mean nothing. It means nothing. You know, in this study now you had that 800 people that were treated over time. Now you start to get real data. And now that study's even been pushed out to people being treated for 4 years with close follow-up. And we understand really what the risks are. And we can understand how to use this medicine clinically and what safety precautions are needed. I don't need to go through that. With all that large data in 2019, in March 2019, the FDA actually granted FDA approval. So where do we stand today? This is the last lesson I want to give to people is just because you get into the red zone, I don't know if people are football fans, but it means if you get within the 20, you're almost there, doesn't mean you're going to score a touchdown. You may get a field goal, or you may get nothing. And the reality is getting even FDA approval doesn't mean that it's going to come to clinical utility. These are the later stages of translational medicine, is you actually have to get just beyond showing that it works, then you actually have to get it into practice and show that it's cost effective and efficient and people can afford it. And the FDA had put what's called a risk evaluation mitigation strategy, or REMS, on the use of ketamine, which makes it very expensive to use. And I think it's an appropriate REMS protocol because it's a drug that has to be used very carefully. But you have to do this, and that really prohibits a lot of people to get access. So there's this big balance between access to treatments, to health equity, and the cost, and how do you figure that out? So with that, we're still at a point where there's still questions left, and we just wrote an international consensus statement looking at where the areas are, and we're still trying to figure out a lot with this. Hopefully that gave you an overview of what it's like to develop a drug from basic all the way to clinical practice, and give you an idea of how many different components there is, including the business component, the regulatory, the political. I mean, there's so much to translational medicine, so many opportunities for you guys to go into besides what you typically may think of as part of basic science in neuroscience. So I'll leave it with that. Hopefully it left some time for questions. Obviously, this is just a small smattering of the people that worked with to get this done. It's a huge number of people, but I just highlighted the people that did some of the really specific studies or part of some of the main papers that I presented. All right, thanks.
So we'd like to open it up to for a few questions for Dr. Sanacora. It's a tradition at Gordon Research Conferences to have students ask the first question. I see a hand up. Yeah.
I just texted my students.
Great. What's your name?
Hi, my name is Miriam Manana, and I'm coming from Regis College. My question is, you did not really talk much about the side effects of ketamine. When you were doing either the preclinical and the clinical research, did you look into what the side effects would be if this was established to patients?
So that's a great question, Miriam. This last slide that I tried to put up here is really looking at the side effects. So preclinically, it's hard to determine side effects preclinically because physiology is different between rodents. The closer you get to non-human, using non-human primate stuff, you can get better, but those become very expensive studies. The unique thing with ketamine was it was FDA approved in 1970. So literally tens of thousands, if not millions of people have already gotten ketamine. I would guess many of you may have gotten it already. It's not uncommon to use it when you have wisdom teeth removed and stuff like that. So it's a drug that had been out there for a long time. So there was a good idea of what the safety was for a single dose. What we didn't know is what if you give it every few weeks? And that really is where it takes this large scale database to get that.
I actually have a question for you, Dr. Sanacora.
Yep.
And it actually stems from one of your first slides about scientific design. I read a commentary recently, I think in Nature, about the replication of bad science. And I'm wondering if you have some thoughts on what we can do as scientists to avoid, we want to replicate our data, but we don't want to replicate science that is poorly designed and poorly executed.
I mean, this is my kind of personal soapbox at this point in my career is how to do this. And it really goes back to, just very quickly, my roommate from college, ended up marrying my housemate from medical school. My roommate from college ended up, he is a PhD material scientist. And my housemate is a basic neuroscience researcher up at the lesser university to the north of us. So I'll say that. And they, we were at dinner and we were talking about our work in neuroscience. And then my roommate for college, who's the material scientist, said, you know the difference between what you do and what we do? It's like we do a study and we get a finding. We replicate it 1,000 times, so we know the variability to 0.001%. It's like you guys do 1,000 studies. If one of them is good, you move on to the next study. And then, but obviously, there's a difference between the fields. You can do a lot of material science studies, replicate them quickly. There's so much control. But I think we do need to take some lesson from that and start to say, we need to, especially preclinical data, you're working in rodents. What's the housing situation like? What's the caging? What drugs you're actually using? How it's given? You have to make it so studies are replicable. It's really a challenge when the NIH has so much money to use. Yeah, and that's the book actually. If you the rigor mortis book, the real criticism is we don't put enough money into the science. So if you're going to do a study, really do a sample size calculation. And it's going to be scary because you're going to see ends of 70, 80. And that's what it takes to get something that is replicable. And the idea of using consort diagrams, which we do clinically all the time. I don't know if people here even know what a consort diagram is, but account for every rodent you use. Because if you publish a paper and say 8 animals, we saw this effect. Well, did you really use 8 animals, or did you do 5 studies to get to that last study where the 8 animals worked? Because you should account for all those animals. If somebody's going to try to replicate it, the chances are they're going to need all those animals again. So it's a fine balance between being very negativistic and trying to highlight where we need to really put our efforts. It's the real challenge in our field is how do we focus our efforts, but yet still support individual thinking that's going to push the field forward.
Thank you. We have other questions for Dr. Sanacora?
Hello, my name is Cindy. I'm from Amherst College.
Hi.
So at the beginning of your presentation, you mentioned how difficult it was to define stress. And then you also mentioned the force swim test with the rodent model. So I was wondering how you decided, or how they decided to pick that specific model to kind of mimic stress, or if there's other models like that.
So the force swim test, the Passault, was developed by Passault decades ago. And the reason that has really continued to be used is that it looks like a good predictor of antidepressant response. But some of it is self-fulfilling because a lot of the antidepressant drugs were developed because it had effects in the forced swim. And early on, they knew the drugs that had TCAs, like mipramine, that had effects changed the forced swim. It doesn't really make sense. Face validity is very low. There isn't much there. There are newer models, and some of the newer models, I'm sure some of you are working with, like the intruder stress model, chronic unpredictable stress model, and then looking at different measures. But it all boils down to the fact that you can't ask a rat or a mouse if they're feeling depressed. I mean, we try to these surrogate measures of despair, of hopelessness. We try to do the best we can. But there's a real question in the field of how valuable these tools actually are. I don't think they're models of depression, but they may be predictors of antidepressant response.
Time for about 2 more.
Okay. Hello, Dr. Sanacora. I'm Lizzie from the Columbia Laboratory College. On your identifying the hazard slide, you really highlighted or foundation hypothesis, and then kind of made a commentary about your experience with that. I was wondering if you could talk more about that, if you don't mind.
So I hope I understood the question correctly. You're asking about the importance of the foundational hypothesis.
Yeah.
Yeah, I would come back to the idea of, science is all about trying to prove yourself wrong, not prove yourself right. I mean, that's the philosophy of science. And I don't think we always follow that so much. So we have a hypothesis, and we tend to do more confirmatory studies than, that's not the philosophy. The philosophy of science is to try to do a study to prove yourself wrong, right? I mean, you've learned that, right? But that not so much what we do. We really try to design studies to prove ourselves right. And I think it really is important to be open-minded and to be critical. So before you move on, before you get too excited about moving on, really do the foundational work to show it's really hard to put a hole in this hypothesis. Or if you have a new mechanism you think work, try to do the study that will prove it wrong, not more studies to prove it right. And that's really hard for us to do because we get excited about it. It's really hard to get funding. And if you shoot your own hypothesis down, then you're really in bad shape. But that's why, the philosophy of science stands here, but the way we're currently moving in our standard way is not necessarily in line with the philosophy of science. So as much as we can do that, I'm a realist. I understand, you have to do both. But before you start to move on, you really want to make sure your foundational hypothesis is solid.
Thank you.
I see a lot of people have questions. In the interest of time, I'm sorry, I think we're going to have to wrap it up and thank Dr. Sanacora for his time. It was an excellent talk.